another protein, which is a checkpoint protein, in response to hydroxyurea or MMS treatment, unlike wild-type cells. So normally when wild-type cells are treated with hydroxyurea, one can detect phosphorylation of RAD53, which is not observed in the MRC1 mutant, indicating that it is required in some way for this modification. MRC1 itself is phosphorylated, and its phosphorylation depends on the ATR homologue MEC1 in budding yeast. And the MEC1 is a kinase, and it phosphorylates proteins at these SQ-TQ cluster domains. And there's a mutant MRC1AQ, which is a phosphorylation defective mutant, isolated in the same lab. This mutant showed compromised viability in the presence of replication stress. And this mutant, which was defective in MEC1 mediated phosphorylation, also failed to activate RAD53 in response to the replication stress, indicating that phosphorylation is somehow important for this signaling. More interesting points about MRC1 also came out from the same study. So these mutants, the phosphorylation defective MRC1 mutants, are replication checkpoint defective, and they fail to activate RAD53 in response to replication stress. But the checkpoint and the replication phenotypes of MRC1 are separable. And the replication initiation is required for MRC1 protein to bind chromatin near the replication origins. Also, as replication proceeds, MRC1 is seen to move along with the replication forks, and the checkpoint activity of MRC1 itself is not required for its localization to the forks. MEC1 is recruited to sites of the DNA replication interface. So MEC1 is the enzyme which phosphorylates MRC1, and indeed it is present at the site of action. MRC1 phosphorylation in response to DNA replication stress is required also for MEC1's accumulation at the stalled fork. So similar approaches were also used to search for mitotic checkpoint defective mutants. There are chemicals such as benomyl or nocordazole or colchicine, which inhibit various aspects of spindle assembly or dynamics, and these chemicals can cause an arrest in M phase prior to metaphase. So Andrew Murray's lab looked for mitotic arrest deficient mutants in budding yeast in the presence of benomyl, and they referred to them as MAD mutants, mitotic arrest deficient mutants. So these are mutants which are sensitive to benomyl and in which the completion of mitosis is not delayed in the presence of benomyl. So these mutants die due to premature exit in the presence of benomyl. And if you look at these mutants under the microscope, you will see that they divide initially more rapidly than wild-type cells to form small microcolonies of 20 to 50 cells. And of course, then they cease to divide. And the cells in these microcolonies show increased frequency of chromosome loss. And from this screen, several such mutants were discovered, which were named as MAD1, MAD2, MAD3, and so on. At the same time, Andy Hoyt's lab also performed a similar screen to look for such mutants. So they describe bug mutants budding uninhibited by benzimidazole. And of course, they again use benomyl, which causes the disruption of the microtubule structure and spindle assembly. And they knew that the arrest in presence of this drug is as a large budded cell with a single nucleus. And in this case, the cytokinesis and the G1 of the next cycle does not proceed, and so they look for mutants where it could actually progress. So they look for mutants where there was new bud emergence, even in the presence of Benomyl, hence the name. And they found a few mutants which fell in this category. These mutants also showed low survival in cortisol containing media, as would be expected. So shown here is the wild type, and when it's treated with Benomyl, it undergoes this kind of arrest where you have the mother cell attached to a medium-sized daughter bud, and it remains arrested in this stage. However, the BUB mutants, the particular mutant shown here is BUB2-2. So this mutant, when Benomyl is added, it actually does not stay arrested as seen for wild type cells, but it keeps on dividing, that is giving off another daughter bud beyond the second bud. So these collection of mutants, the BUB mutants, also were likely to be defective in a checkpoint that monitors the spindle assembly. So these findings led to the discovery of components of the spindle assembly checkpoint. The spindle assembly checkpoint monitors whether assembly of the spindle is complete or not. That is, have all the sister chromatid pairs attached to the microtubules of the spindle or not. So unattached kinetochores lead to the assembly of the mitotic checkpoint complex shown here. So at the top you can see a spindle where the assembly is incomplete. One pair of sister chromatids has one unattached kinetochore. It has not yet attached on both ends, whereas the other chromosomes are attached. So this is an example of an incomplete spindle assembly, whereas the bottom one is in metaphase, where all the chromosomes are at the equatorial plane and they have, each of these pairs have attached to the spindle microtubules coming from opposite poles, which is when the cell cycle would progress. So unattached kinetochores, they lead to the assembly of this complex. So as long as there's even one unattached kinetochore, you have this mitotic checkpoint complex active, and its role is that it inhibits APC, the anaphase promoting complex, which is a ubiquitin E3 ligase complex that I mentioned earlier. And one of the subunits of this complex is CDC20. So when all the chromosomes are attached and aligned, this inhibitory complex is no longer formed. And so this weight anaphase signal is extinguished. And CDC20, which was part of this complex, is a subunit of APC. So now it can go and it binds APC. And APC now is active and it can ubiquitinate its targets. Two important targets of APC are cyclin B and securin, also known as PDS1 in budding yeast. So these proteins undergo ubiquitination and ubiquitin-mediated degradation by the proteasome. And upon the cyclin B destruction, the CDK1, which was active, the mitotic CDK, is inactivated. Upon securin destruction, securin itself was in a complex with a protease known as separase. So when securin is destroyed, separase is set free. And it's a protease whose target is the cohesin complex. So it goes and cleaves one of the subunits of the cohesin complex. The cohesin complex holds sister chromatids together. It brings about sister chromatid cohesion. So When cohesin is degraded, then these two sister chromatids, they can come apart and the metaphase to anaphase transition occurs. To summarize the checkpoints that I discussed today, the DNA damage checkpoints operate in interphase. They operate in G1 phase, in S phase as well as G2 phase. And these checkpoints, they slow down progression of the cell cycle to allow for repair of the DNA damage. So as long as DNA damage is there, the cell cycle is stopped, and when the damage has been repaired, then the checkpoint is inactivated and then the cell cycle can progress forward. Mitotic checkpoints also monitor complex events in mitosis. So there are multiple mitotic checkpoints, but today we have discussed a little bit about the spindle assembly checkpoint. The spindle assembly checkpoint checks for the proper attachment of chromosomes to the spindle, and it provides time for all the chromosomes to attach to the spindle microtubules. If the attachment is incomplete, this checkpoint is active and it prevents cell cycle progression. It prevents the metaphase to anaphase transition. And when the spindle assembly is complete, the checkpoint is inactivated and the cell cycle can progress forward. To summarize, checkpoints ensure that the cell cycle does not proceed under potentially dangerous conditions. Checkpoints require a mechanism to detect errors or problems in a cellular process via a sensor. They require a transducer of this signal and an effector that generates a reversible signal that inhibits cell cycle progression. Checkpoints generally slow down or arrest cell cycle progression to enable cells to fix the damage before proceeding further. Checkpoint mechanisms may be dispensable for a given cell division or for viability, but they are critical for maintaining the fidelity. Mutations in checkpoint genes have been reported to be associated with cancer predisposition and with progression of cancer. And sometimes if a cell is unable to fix the damage, it may undergo apoptosis, that is a kind of a cell death that removes that defective cell from the milieu. External stimuli such as nutrient availability or certain factors such as mitogens can affect the entry into the cell cycle by stimulating the activities of specific CDKs. So these also provide a point in the cell cycle on which control can be exerted. Thank you. Hello everyone, I'm Shikha Laloria, Professor of Biochemistry at IISc. Today's lecture is on the ubiquitin proteasome system, that is an intracellular protein quality control system present in cells. It is involved in the degradation of proteins in the cell. We will also discuss the complexity of the system and also about ubiquitin and ubiquitin-like proteins and how the post-translational modification by ubiquitin and ubiquitin-related proteins impacts several cellular processes. The ubiquitin proteasome system is a highly complex system for the post-translational modification of protein targets by ubiquitin and for their controlled degradation. This complexity allows for possibilities of regulation at multiple levels, and this depending on the cellular conditions, and also for specifically modulating the pathway for the desired outcomes. It's one of the main cellular quality control system for proteins inside the cell. And the requirements for this system to function are a polyubiquitin tag has to be attached to the proteins that are destined for degradation. There is a complex multi-component nanomachine, the proteasome, that degrades these tagged proteins. There are also many other regulators, enzymes, and adapters for bringing about the specific recognition and modification of the target protein at the appropriate time, and also for its recognition by the proteasome and for degradation. And of course, this process requires energy in the form of ATP. In addition, ubiquitination, which is also often referred to as ubiquitination, can result in effects other than degradation. Sometimes the polyubiquitin chain is not assembled, but the protein can either be mono or multi-ubiquitinated. And such modifications by ubiquitin and the ubiquitin-like proteins can alter the properties of the target protein in terms of its activity or its interactions with other proteins, or its localization in the cell, or impact its function in various ways. So this provides additional levels of complexity and regulation. Ubiquitin is a short 76 amino acid polypeptide that is covalently conjugated to the target proteins. Ubiquitin is attached via its own C-terminus to the amino group of the side chain of lysine residues of the target substrate protein via an isopeptide linkage. The carboxylic group of ubiquitin is attached to the amino group of lysine side chain rather than to the alpha amino group. The conjugation with ubiquitin can alter the fate or function of the modified protein. As I mentioned, it may get targeted for degradation, or it may also alter its properties and interactions. There are many other ubiquitin-like polypeptides, and these can also be conjugated to the target proteins, resulting in reverse outcomes. And ubiquitin, though, is the founding member of this family of modifiers, which bear some structural resemblance to each other. So the ubiquitin-like protein family is a family of small proteins which is related to ubiquitin. So these are all known as ubiquitin-like proteins, or UBLs, and they share a similarity in 3D structure. They all have a characteristic ubiquitin fold, or also known as a beta grass fold. It consists of a 5-stranded beta sheet, an alpha helix, and a short 310 helix. This 310 helix, it's a variation from the alpha helix, where it has a repeat unit comprising of 3 amino acids in one turn. The mature form of the ubiquitin-like polypeptide ends with a signature diglycine sequence. And this is often exposed by processing of a ubiquitin precursor polypeptide. The carboxyl group of the C-terminal glycine is the site of attachment to the substrates, and the lysine side chains are the most common target sites within substrate proteins, resulting in an amide or isopeptide bond between the ubiquitin-like protein and the substrate. Of course, there are always exceptions in biology. So there are rare examples of conjugation of the ubiquitin C-terminus to a free alpha amino group of an N-terminal residue of a protein, or even via cysteine side chain, for example, in the MHC class 1 heavy chain, in a reaction that is catalyzed by a viral E3 known as MER1. So exceptions do occur, but mostly the attachment is to the epsilon amino group of a lysine side chain. The ubiquitin-like proteins are highly conserved. That is, for example, there are only 3 differences in amino acid sequence out of 76 positions between yeast and if you compare it to the human ubiquitin. Ubiquitination is a very complex multi-step process, and here I'm trying to show a generalized ubiquitin conjugation pathway. Now, of course, the first step, which is not shown here, is that there are precursor ubiquitin-like proteins, and they are processed by DUBs, that are deubiquitinating enzymes, or ULP, ubiquitin-like protein-specific proteases, to expose this C-terminal glycine in the mature ubiquitin. That's not shown here, but by that cleavage you would get a ubiquitin polypeptide whose C-terminus is a glycine. Now this processed ubiquitin can be conjugated and cannot bind an